which is probably the most important and most underestimated step in high dimensional data analysis. So flow data, because of the techniques, approximate a log normal distribution. Don't be afraid of this. It means that if you apply a log-like transformation to your data, log, the exponential or whatever, your data will look better, will look more normally, almost normally distributed. This is why we always apply a scale when we open plots and histograms. Okay, we do it by default, basically. We do not even think about that. The reason for this is that the variance of the data, of the raw data that the instrument acquire, increases with the increasing of intensity. So as you can see here, this is a non-scaled, you can also say linearly scaled. It's the same stuff. Linearly scaled or unscaled is the same. This is an unscaled data. This is a flow cytometry CD8 APC cyclone 7. So this is unscaled, right? This is the raw data. You see that the negative, it's really compressed. The positive, it's really broad. And this broadness increases with the increase of intensity. So if you have a peak at 50,000, it will be even broader. Okay, and so on and so on and so on. The more you go right, the broader will be the peak. And I mean, we, okay, flow cytometrists, humans, knows that this is one peak and this is another peak. So because we know biology, the algorithms usually rely on distances. So if you ask an algorithm, looks, can you find for me two populations by looking at this data? Well, the algorithms will probably draw some wrong conclusion, because think about analyzing cell A, B, and C. The distance between cell A and B, it's 10,000. The distance between B and C, it's 20. So for the algorithm, which is relying on distances, A and B are more similar than B and C. So it will probably do something like this: A and B together, C apart. And this is not what we want, right? Because we know that it's wrong. So if you apply a scale, you're basically transforming your data. You will get different values, different numbers. It's like creating it's like mapping the data into a new sort of scale. And the idea is that you want to shrink the distances at higher value. So instead of having this peak broad like this, you will have something like this, which is now more comparable. So this is the negative and this is the positive. This is exactly how we are habit to look at our data. And now, if you ask the same algorithm as before, please do the same as you did before, but using the scale data. Now the algorithms will group B and C, because between B and C you have a distance, which is 2, which is smaller, than the distance between A and B. So now A, it's apart, and B and C are together. So overall, what you're doing, it's making the distances at higher value, giving the distances at higher value the same importance of the distances at lower value. You are balancing, okay, the importance of distances. And graphically speaking, all what you have is that the two peaks, the negative and the positive, are more comparable, graphically more comparable. This is all what you want. You are basically making a favor to the algorithm. Exactly the same, it's happening, of course, in 2 dimension, 3 dimension, 4 dimension, and so on. So this is just an example. This is the same, our old friend CD8 APCH7. And this is CD4 P cyclone 7. Now, if you look at this, and if you do not know biology, just look at this. It seems that this light blue, by looking at CD4 P cyclone 7, this light blue, it's closer to the red one, okay, than to the green one. And if you look at the y-axis, CD8, the light blue, it's closer to the green one than to the red one. So if you look at this data, you may say, well, this is a double negative. Okay, but of course, you know it's wrong. How to help the algorithm avoiding to draw some wrong conclusion? You apply the scale. And now for the algorithms, there is no doubt. The blue and the red are close, closer than the blue and the green. The blue and the green are closer than the blue and the red. And now the blue one, it's clearly a double positive. So this is what you're doing when you apply a scale to parameters. There are a lot of scale available. Linear, which basically means do nothing. Be exponential, hyperlog, logarithmic, blah, blah, blah. It's not really a matter of which scale you're going to use, because most of the scale can help you in reaching the same, almost the same results. It's a matter of finding a scale that fulfill these two requirements. And the first requirement is what we said before. So making distances of equal importance allowing the range of values. And the second is, it's good to find a scale that can handle negative values. This is the reason why nowadays we're no longer using logarithmic scale, okay, that was used to use in the old ages, because logarithmic scale cannot handle negative values, or basically you are losing information. So let's remove logarithmic scale. You can still use it, for example, if you're dealing with like for a scatter size scatter, because you're not expecting negative events. But for fluorescence, basically, you are just using hybrid scale. We will see what an hybrid scale is in a few seconds. You see here, I have added an asterisk. So most of the algorithms, most of the downstream algorithms benefit from a scaling because of the reason I mentioned before, right? So you want to make distances more homogeneous. There are some exceptions. One exception is, for example, and it's not related to high-dimensional data analysis. One exception is, for example, algorithms that fit DNA cell cycle data. Okay, so algorithms that analyze DNA cell cycle data, so G1, S, and G2, usually want the data to be linearly scaled. Okay, another exception is, for example, FlowAI. Okay, FlowAI is suggested to be used on linear scale, so on raw data. Okay, FlowCut, for example, it's suggested to be used on scale data. So there are a few exceptions. From the top of my head, I can think about DNA cell cycle and FlowAI, but it might come an algorithm in the near future that says this algorithm should be run on unscaled data. So be prepared for that. Always ask yourself, shall I scale the data before running the algorithm of interest? 99% of the time, the answer is yes, but always be prepared. So read the original paper, Google on internet, or send us an email if you have any doubt. Log scale, there is a question. Log scale do not change the numerical value. Then how come scaling change the value for algorithm? So thanks for asking. There is just one difference between how you scale the data on a plot just for visualization purposes and how you scale the data for an algorithm. When you're scaling the data for visualization purposes, like for example, in this case, on the y-axis, you are scaling the data, but still on the axis, you are showing the raw values, okay? It's just a visualization, it's just a trick. When you're doing scaling for algorithms, algorithms do not rely on the aesthetics, okay? Algorithms run on the raw data. So if you log scale a value at 10 to the 5th with base 10, for the algorithm will be 5, okay? Will not be 10 to the 5th. This is the only difference. So we rely on the aesthetics, algorithms rely on the actual number. And what I showed you here, let me zoom in, those are the actual number that algorithms will use. What algorithm name is the comp and scaled sample applying? Oh, in this case, sorry, in this case, I don't want to tell you which scale I used, okay? Because I don't want to tell you use this one. This is just one of the scale available in flow cytometry. This can be by exponential, this can be hyperlog, this can be arcsine. I will show you this in a minute. So it's not really a matter of which scale, it's a matter of which scale and which setting, how you set that scale, because with different scale, you can achieve the same results. So which scale shall we use? Not log, unless you're dealing with some data that doesn't have any negative values. Usually use hybrid scale. So be exponential, hyperlog, arcsine or lean log. They all do the same stuff basically in a mathematically different ways, but they are all doing the same stuff. So what they're doing is transforming the data with a mixture of linear and log scale. This is the trick, okay, we can use to also plot negative events. So around 0, all those scales use linear, and far from the 0, those scales use logarithmic or some slightly different variance. Okay, but the idea is that you have a logarithmic around 0 and then something else like, sorry, linear around 0 and then something else afterwards. Okay, for example, b exponential is a mixture of linear and logarithmic. Hyperlog basically doing the same. Arcsine H is slightly different. Lean log, it's really linear and logarithmic. So all what you need to keep in mind is that it's a mixture of scale, and around 0 it's linear, and above it's above this threshold, okay, it's something different. Anyway, you're doing something that compress the positive and still allows you to have a good resolution in the negative for the negative peak. The point in which the scale moves from linear to something else, it's called transition point for the hyperlog and the lean log. It's called below 0 parameter for the b exponential. It's called cofactor for the arcsine H. And again, it's not really a matter of which one you use. Of course, if you try all of them, you will find some differences. Maybe someone works better than the other, but they all do the same stuff, okay? They compress the positive in a way that you are making more homogeneous the distances. This is a comparison, okay? It's worth looking at this slide. This is the same data set, the same parameter, our old friend CD8 APC Cyclone 7, scaled with b exponential, hyperlog, and arcsine H, with different transition point, okay? 100, 500, 1000, 5000. So basically the linear scale in this case extend up to 100. In this case extend up to 500, up to 1000, up to 5000. So if you look at this, you can see that, for example, all those three, b exponential, hyperlog, arcsine H with this transition point are not really good, because the negative peak, it's not really visible. Like, you see how many events are stacked on the axis. This is too broad as compared to the positive. This seems to have like a 2 peaks. Okay, so I will not go with any of them. Similar stuff with this one. Okay, this is really compressed, this is really broad, this is really compressed, this is really broad, and so on. So probably something in between will be fine. And you see that it doesn't really matter which one you go with, the exponential, hyperlog, or arcsine H. Like this one, it's very similar to this one, it's very similar to this one. Okay, so stick with the one you want. In a few slides, you will see that b exponential gives you some advantages, but up to now, you see it's not really a matter of which scale, it's a matter of how you set that scale, because you can achieve the same result. This is why when you ask which scale you are using in this picture, I can tell you in this case was b exponential, but you see I can achieve the same results also with other scales. Okay, so questions. Wow, Andrea, are you telling me that I should compare all scales, all below 0 and transition points for all parameters in my files? I know I might have like 50 parameters for all the data, file by file. It will take forever. So in principle, yes, because otherwise, how can you know which scale to apply? You cannot apply the same scale with the same transition point for all parameters and for all files. So in principle, yes. In the real life, there are tricks thanks to the B exponential scale that will make your life much, much, much easier. But I want to go through this process, this manual process, this one, with you a little bit, like 5 minutes dedicated to do something manually to then appreciate much better the automatic way. So let me open a layout in SGS Express. In this layout, let me close this. In this layout, I have a file. Actually, I have 4 files here. I'm just analyzing file number 1. I have a gate of interest, and I want to decide which scale to apply to the cells within the gate of interest. Okay, if you change the gate, if you change the file, the scales you might need will be different. So let me open an histogram. Okay, let's do it manually. I'm going to create a pipeline. I'm going to add a downsampling gate, downsampling step, and I want to gate on gate 1. Okay, and I'm going to add just one single step, which is the scaling, mathematical scaling step. So in the meantime, there is a question: if you have inconsistency in staining intensity between samples, can scaling help make sample files more comparable for downstream tSNE UMAP? Not really, not really. So you can account for the spread of the data. If you have for example, a negative or a positive which is more spreaded than the other one, you can do something with the scaling. But if you're speaking about the intensity, scaling will not really be helpful. Like, think about scaling with log scale, which is the simplest one. Like, in one sample, your positive it's 10 to the 4, log base 10 will be 4. In other sample, you have 10 to the 5th, log base 10 will be 5. So still you will have a discrepancy. So this is something that if you want, you can do with the normalization to the minimum maximum, for example, maybe normalization to the mean. So it's something that we will see tomorrow. Thanks for asking. Okay, so I've created a pipeline with a gate step here, gate 1, a scaling step. I want to scale CD45. I want to scale it with B exponential, below 0 parameter, manually defined to 200. So CD45 array. Let me apply the pipeline and let me automatically execute it. So now I have the CD45 array scaled. You see that the values are different. So do you like it? I don't, because you see the negative, it's not really visible. So I will try to increase the below 0 to 1000. Oh, wow, much better. So this is BEXP, and we have set it to 1000. If you try increase it, for example, like 5000, you will see the negative will be more compressed than the positive. We want some balance, right? Just for fun, let's do it with arcsine H. Here we go, arcsine H. Definitely bad. Arcsine H cofactor 5. You see the negative, it's splitted by 2. Doesn't make sense. 100. Still the negative splitted by 2. Sorry. 5000 too much. 500. Whoa, okay, 500. You see different scale require different adjustment. Hyperlog. Last one. Hyperlog. 200 definitely not good. I cannot even see the negative peak. 1000. Okay, 1000. Okay, then of course, if you said 1000 or 900, it will probably make a small difference. So you can also write here 900, that's fine. We just want to avoid very bad stuff. And then live and dead. Live and dead is tricky in this case because I cannot really see a clear positive peak. So let me switch to a 2D plot. The positive peak, it's this one. Sorry, the positive population is this one. You see how broad is the negative? This is hyperlog 900. Okay, live and dead. 900, it's not enough. So 5000. Oh, this is 50,000. 550, sorry. 5,000. Okay, much better. So 5,000. How about arcsign H? No. Better. And how about the exponential? Okay, not bad. Maybe we can also go higher. Okay, 10,000. So you see, different scale require different setting, and different parameters require different setting. But more or less, you can achieve the same. Okay, like the exponential 1000, arcsign H 500, hyperlog 900 were really similar. Now the question, shall I do this for every single parameters, every single file? Likely, the B exponential scale is able to automatically find the below zero parameter. So what you see on the left are 4 different manually defined below zero parameter for the CD8 APC size 7. What you see on the right is the automatic below zero, the distribution that you will find if you use the automatic detection of the below zero parameter. Is there any quantitative way to choose transition value, or just highballing? It's fine. Most of the time, your highs are fine. So there is a little bit of tolerance. Like if you use 500 or 600 or 700, probably there will not be so much difference in this case. Okay, you see between 500 and 1000, yes, the negative, it's a little bit more compressed. That's fine. Just stay away of like 100 and 5000. Okay, but those are just value for this parameter, this data set. Okay, if you use another data set, another sample, you might find the best value will be like 20,000. That's fine. So you have two ways. The B exponential, the B exponential scale has two ways of calculating, two methods of calculating the below zero parameter. One is called BD fax diva, one is called fifth percentile. The second one, it's less affected by outliers. And I want to show you how to use it, and I want to show you what's the difference between BD fax diva and fifth percentile. So let me remove this. Let me go back to our histogram. Now what I'm going to do is scaling. We can scale all of them all together. In this case, I don't really care about scaling scatters. You can scale scatter, of course, with the exponential. In this case, I just want to focus on parameters. So in my scaling step, I'm scaling everything with B exponential, and I'm asking FCS Express to automatically calculate the below zero parameter using the BD fax diva method. So execute. How can I view the results of this scaling? Well, once you have opened an histogram, you can go to multiplots and create a multiple histogram. Now, this is the list of, clear selection. This is the list of all unscaled and scaled parameter. I'm just interested in the scaled one. So I can filter, I can select all, and I can view all of them in a multiplot. Okay, so what you see here, it's a bunch of histogram displaying all the selected and scaled parameter. This is the result of the B exponential scaling with BD fax diva method automatic detection of the below zero parameter. You see that it works pretty well. Okay, you have peak. In this case, you cannot really see that peak. So I will show you. You can still put your hands and change something specifically, but most of the time you see that positive and negative peak are really well homogeneous. Sometimes it's hard, like in this case. When you do not have two peaks, the only thing you can do is like guessing the best below zero parameter, or simply like if you have a sample in which you have two peaks, you can calculate the below zero parameter on that sample and use the same below zero parameter for every other sample. Okay, so every time you need to do the best you can. You cannot do any miracles. Also here, you see, you don't have any peaks, so there is nothing you can do unless you have a sample with two peaks. So this is just one sample. Okay, let's see whether sample 2 also is scaled properly with this. Sample 2 is going to be loaded and scaled. This is like alpha million data points. Here we go. Now we are also screen sharing, so it's not the best scenario. It's fine. You see, also for sample 2. So the idea is that you will check all of them and see whether it's fine. If you change the gate, okay, if you change the data set, it might happen that you find out that this scaling is not appropriate for all parameters. Maybe some parameter doesn't scale well if you change the gate because you're changing the data set. So let me, for example, switch off the gate downsampling and let me scale dataset 2 on whole files. And I want to show you that if you let the B exponential scale calculating automatically the below zero parameter on the whole data set, you have one parameter that doesn't scale well. So we are scaling with automatically calculated below zero parameter using the BD-FAX-DIVA method. This happens, let me show it here for you, CD3 scaled. This is happening because there are some very, very, very rare outliers, okay, in the negative range. Now it's no longer negative. So if you want something more robust, that it's not so affected by outlier, you can switch the 5th percentile method. And you will see that the CD3 will be scaled in a much more meaningful way. You see, now we are no longer being biased by outlier, and the CD3 scaled, it's scaled properly, okay? Now, what happens if you want to change specifically one sample? Let's just take one before concluding. Let's say that we are happy with all of our parameter. We're happy with the below zero parameter calculated with the 5th percentile, okay? Except for, there is nothing really bad here, but let's say that we want to change this one, okay? We want to make this negative more compact. It's not really required, and just finding some let's say parameter to tweak manually. Let's say that we want to change this CD20 and that we are not happy with the automatic below zero detection. So I will not scale the CD20 here. Okay. I will add a new scaling step to my pipeline. Yes. And this will be only for CD20. So in this scaling step, I will specifically scale the CD20. Where is CD20? CD20. Let me filter. Okay. How I want to scale it? Well, manually. Let's say below zero, set to 10,000. Let's try. Okay. So now it's going to be calculated, and this is the CD20 scaled. Okay, if 10,000 is not enough, let's say 20. And you can keep this automatically run pipeline checked if you want. I'm happy to keep it off, but sometimes it's useful. So you see, now our CD20, it's more compact with the negative, and it's really, let's say, comparable to the positive peak. Okay, all what I've done is scaling everything else automatically and just scaling the CD20 manually. Is this okay for every other file? Let's see. Let's see file 1. Now file 1 is being loaded. Here we go. It's fine. So most of the time, you will not need to let's say, manually adjust any parameter, but it's useful to know that you can do that. So it's a way, again, scaling, it's a way to make positive and negative more comparable in terms of spreading. And XGS Express allows you, with the pipeline and with the B exponential scale that can automatically detect the below zero parameter, in finding the best scale parameter by parameter. So think about how much time you're saving doing this instead of doing this manually one by one. Okay, time is running. I want to conclude. Here is the last slide. So about scaling, we know that the variance of the flow data increases with increasing of intensity, and we need to take some action to correct this because algorithms rely on distances. Most mathematical algorithms assume that the distances are comparable along the range of data. Scaling, it's a way to shrink the point-to-point distance at higher value to make them more comparable to the distances at lower value. And most of the algorithms benefit from this. Okay, there are a few exceptions. We mentioned two. I cannot think of any other exception. One, it's DNA cell cycle data analysis. One, it's Flow AI. So this is the last slide. Tomorrow we will address as a first topic data normalization, and as a second topic tomorrow we'll focus on downsampling. And you will see there is much more to learn than the very frequently used random sampling. So if there is any question, this is the right time to ask. I can stay here for a few more minutes. If there are no questions, I hope you will be able to join tomorrow for the second part of this series. So I think I've answered all the questions in the list. Okay. So thanks to all of you. It has been a pleasure. Thanks for asking so many questions. See you tomorrow.